ancient DNA research has transformed our understanding of human prehistory over the past 2 decades. Scientists have successfully sequenced genomes from Ice Age Europeans, ancient Anatolian farmers, Bronze Age steppe herders, and Neolithic populations from across Eurasia and the Americas. These genetic snapshots have rewritten migration maps, overturned long-held theories about population movement, and revealed entire chapters of human history that left no other trace. The field has been one of the most exciting frontiers in science. But there was always one glaring blind spot. Hot, arid environments destroyed ancient DNA with brutal efficiency. The chemistry is straightforward and merciless. Heat accelerates the chemical reactions that break down genetic material. Temperature swings between scorching days and cold nights shatter DNA strands at the molecular level, fragmenting them into pieces too small and too degraded to reassemble into anything readable. Decades of attempts to sequence genomes from North African archaeological sites, mummies and skeletal remains from Libya, Egypt, Morocco, and Algeria had produced nothing. The consensus in the field was not merely pessimistic. It was categorical. You cannot recover whole genomes from remains preserved in hot desert conditions. Full stop. This is the wall that archaeogeneticist Nadia Latova-Salem and her colleagues at the Max Planck Institute decided to walk straight into. Their reasoning was based on one key biological insight. While DNA in most of the body degrades rapidly in harsh conditions, the densest structures in the human skeleton, the teeth and the inner ear bone known as the petrous bone, offer a degree of natural protection. These mineralized fortresses shelter genetic material at higher concentrations than anywhere else in the body. They are the last place ancient DNA survives. Salem's team extracted material from the Takarkori mummy's teeth and leg bones, then applied the most advanced next-generation sequencing techniques available, working methodically through fragments that the desert had spent thousands of years disassembling. Strand by strand, piece by piece, they rebuilt something the Sahara had tried its best to erase. They recovered 2 complete genomes, both of them. When Johannes Krauss, senior author of the resulting study and director at the Max Planck Institute, reviewed the final sequencing data, he reportedly sat in silence for an extended period before saying anything at all. These samples, he eventually noted, come from some of the oldest mummies in the world for which complete genomes have been recovered. The technical achievement alone was historic. But what the genomes actually contained was something far more significant than any laboratory milestone. The first thing Salem's team did with the recovered genomes was what any geneticist would do: compare them against everything else in the record. They ran the Takarkori DNA against a reference panel of 795 present-day genomes and 117 ancient genomes drawn from populations across Africa, southwestern Asia, and Europe. The goal was to place these 2 women on the human family tree, to find their relatives, to understand where they came from and who they were connected to. The African humid period should have made this straightforward, with the Sahara transformed into a continuous green corridor connecting North Africa to sub-Saharan Africa. Gene flow between northern and southern populations should have been natural and essentially inevitable. The expectation was clear. The Takarkori women would carry genetic signatures reflecting that connectivity, showing ancestry shared with populations living to their south. They carried none. 0 sub-Saharan African ancestry. Not a fragment, not a statistical trace, not a hint. These 2 women, living at the heart of the most biologically connected era the Sahara had ever known, during a period when there were no physical barriers between them and populations hundreds of kilometers in every direction, had maintained complete genetic separation from the world around them. The Green Sahara, which by all logic should have been a highway for human genetic exchange, had functioned for their ancestors as an invisible wall. Krauss ran the comparison 3 times. The result did not change. The isolation was total, and it was deep, far deeper than the 7,000 years separating these women from the present. When the team dug further back, searching for any known ancient population that shared a meaningful genetic connection with the Takarkori women, they found something that made the strangeness even stranger. The closest genetic relatives were not nearby populations. They were not contemporary North Africans. They were not Mediterranean groups or Nile Valley communities. The closest match in the entire ancient DNA record was a population of hunter-gatherers buried at Grotte de Pigeons in Tafril, Morocco. Individuals who had lived approximately 15,000 years ago, thousands of years before the Green Sahara even began, in a completely different geographic region, in an entirely different ecological world. And yet the genetic signal connecting these two populations was clear and specific, a deep shared ancestry found nowhere else in the available record. Not in Europe, not in the Near East, not in sub-Saharan Africa, not in Central Asia. Nowhere. What this suggested was extraordinary. Stretching along the northern edge of the African continent for a period spanning tens of thousands of years, there had existed a distinct human lineage, a population group carrying a specific ancestral signature that appeared in no other living or ancient population ever studied. Population geneticists had actually suspected something like this might exist. For years, subtle statistical anomalies had appeared in the genomes of modern North African populations, faint traces of an unidentified ancestral contribution that did not correspond to any documented group. The signals were too consistent to dismiss and too vague to prove. Researchers had given the theoretical source of these signals a name before any physical evidence was found. They called it a ghost population. The Takarkori mummies gave that ghost a body. In the summer of 2023, inside a climate-controlled laboratory at the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, a team of geneticists sat staring at computer screens in complete silence. What they were looking at was not supposed to exist. The data on those screens represented something the entire scientific community had declared impossible: a fully sequenced genome extracted from a mummy that had spent 7 millennia baking in the Sahara Desert. But the impossibility did not stop there, because what that genome said about human history was something no textbook had ever prepared anyone to read. This is the story of the Takarkori mummies, 2 women who died in what is now southwestern Libya roughly 7,000 years ago, and whose preserved remains quietly waited beneath layers of rock and sand until modern science finally had the tools to hear what they were trying to say. What those women's DNA revealed did not just fill a gap in human history. It tore open a chapter that nobody even knew was missing. To understand why this discovery matters so profoundly, you first need to understand the world these women lived in. The Sahara Desert today is one of the most hostile environments on Earth, a vast, scorching expanse covering roughly 9.2 million square kilometers, stretching across the entire northern breadth of the African continent. It is a place of extreme temperatures, near zero rainfall, and almost complete biological silence. Nothing about it suggests it could ever have cradled human civilization. But between approximately 14,500 and 5,000 years ago, the Sahara was something else entirely. Scientists refer to this era as the African Humid Period, a prolonged climatic shift driven by changes in Earth's orbital cycle that redirected monsoon rainfall deep into the heart of North Africa. The results were transformative. Permanent lakes formed across what is now empty desert. Rivers carved their way through landscapes that today hold nothing but dunes. Grasslands stretched from the Mediterranean coastline down toward the Congo Basin. Elephants, hippos, giraffes, and crocodiles populated a lush, green world that would be utterly unrecognizable to anyone who knows the Sahara today. And people thrived it. Rock art found across Libya, Algeria, Chad, and Niger depicts this lost world in extraordinary detail. Cattle swimming in lakes, hunters pursuing game across open plains, families gathered around water sources. These were not wandering bands simply passing through. These were communities with roots. The Takarkori rock shelter itself, located in the Fezzan region of southwestern Libya, shows evidence of continuous human occupation stretching back more than 8,000 years. 15 individuals were buried there across generations, mostly women and children, alongside pottery, animal bones, and trade goods that speak to a society with structure, memory, and permanence. 2 of those individuals never decomposed. The dry desert air mummified their bodies naturally, preserving skin, ligaments, and soft tissue in a state of remarkable integrity. When Italian archaeologist Savino di Lernia first uncovered their remains during excavations at the site, he described his hands shaking. He knew immediately what he was looking at. The question that would take years to answer was what they were carrying inside them. The team did not stop at 15,000 years. They pushed further back, testing the Takarkori genomes against some of the oldest sequenced human remains in the archaeological record. 50,000-year-old individuals recovered from Zlatý Koun in the Czech Republic. These ancient people represent one of the earliest known waves of anatomically modern humans to migrate out of Africa, the original dispersal event that eventually populated the entire planet beyond the African continent. The result stopped the room. The Takarkori women were more closely related to these 50,000-year-old individuals than to any present-day sub-Saharan African population. Their ancestry traced in part to that original out-of-Africa migration, the very first chapter of the human story beyond the continent. And then, at some point after that initial dispersal, their ancestors had either returned to North Africa or had remained behind in a way that left them genetically anchored to that founding population, isolated from the subsequent waves of mixing and migration that reshaped every other human group on Earth. The scale of this isolation is genuinely difficult to comprehend. 50,000 years. While that timeline was unfolding, the rest of humanity spread across every inhabited continent, split into thousands of distinct language groups, invented agriculture, built the first cities, developed writing, constructed empires, collapsed them, and built new ones. Ice ages came and receded. Sea levels rose and fell by 100 meters. Entire megafaunal species, woolly mammoths, cave lions, giant ground sloths, appeared, dominated their ecosystems, and went extinct. The Takarkori lineage persisted through all of it, genetically unchanged, genetically separate, in the same corner of the same continent. Perhaps the most intellectually destabilizing aspect of the Takarkori discovery is not the genetic isolation itself, but what that isolation reveals about how human civilization actually spreads. The women in the Takarkori shelter were pastoralists. They herded domesticated animals. They produced pottery. They participated in long-distance trade networks that connected them to communities across sub-Saharan Africa and the Nile Valley. Their material culture was rich, diverse, and clearly informed by contact with the broader ancient world. Pastoralism, the practice of herding domesticated animals like sheep, goats, and cattle, is generally understood to have originated in the Fertile Crescent of the Near East, where these animals were first domesticated roughly 10,000 years ago. The traditional model holds that this technology spread outward from its origin point through migration, herding communities physically moving into new territories and bringing their practices, their animals, and their genes with them. Technology travels in people. People carry their DNA. The Takarkori genomes contain no Near Eastern ancestry, no genetic signatures from Levantine populations, no Anatolian farmer DNA, no trace of any population that the conventional migration model requires to explain how herding reached the Central Sahara. These women were practicing a technology that supposedly arrived via migration from people.